Today we'll discuss about the structures of protein and their conformation and the reason behind these conformations. As we all know that to make a molecule from their unit structure, chemical bonds play a very important role. In case of protein, these bonds are covalent and non-covalent. Peptide bond, which is a type of covalent bond, play a very important role in the formation of a polypeptide or protein from amino acids. Now let's discuss in brief about these important bonds who give rise to the formation of different structure of protein. So here, suppose we have two amino acids which are come together to form a peptide bond, and in doing so, one water molecule released out. Thus, the reaction is called as condensation reaction. And since the nitrogen of this amino acid is with lone electron pair who attacks the carbon of this acyl group to form a peptide link or amide linkage. Thus, the reaction is called as nucleophilic acyl substitution reaction. This leads to the formation of a peptide linkage or amide linkage or say amide bond, as it is formed between the carboxyl group and the amino group. Thus, it looks like CONH is involved in the formation of this peptide linkage. So it is also called as amide bond or amide linkage. And if and in the same way, if other amino acids also link to each other in a series, this gives rise to the formation of a long polypeptide chain with amino terminus at left side with a free amino group and carboxyl terminus or at other end with a free carboxyl group. This peptide bond possesses a partial double bond feature that restricts the motion of the molecule around it and makes all the 6 atoms, that is carbon, oxygen, nitrogen, hydrogen and carbon, in a rigid plane. So how does the protein get folded into different configuration to give rise different structure? Answer is dihedral angle and torsion angle that is present between N, C alpha and carbon alpha. The angle between N and C alpha is represented by phi, while the angle between carbon and carbon alpha is represented by psi. And the degree of the rotation of these both angle is from minus 180 to plus 180. And these are the main angle who give rise to different conformation of protein due to their motion or can say rotation around these angles. So let's discuss about the structure now. What is primary structure? These are the simple linear polypeptide. It looks like a group of amino acids standing in a single line and bonded via peptide bond or amide bond. When the dihedral angle within the primary structure changes their configuration, it gives rise to a secondary structure, which could be helical, sheet type or turn. Here we have a ribbon-like structure. Suppose it's a protein. When some torsion angle shows their movement, this becomes an opportunity for other atoms like hydrogen of amino and carbonyl oxygen to come together to form a hydrogen bond to give rise an alpha helix. In case of alpha helix particularly, the hydrogen bond is formed between the first amino acid and the fourth amino acid. And in the same way, all the amino acids are hydrogen bond linked to form an alpha helix, which is a right-handed structure. This hydrogen bond is that forms along the alpha helix is parallel to the axis of this helix, with each turn is at 5.4 Armstrong apart, including 3.6 amino acids, and each amino acid is 1.5 Armstrong apart from each other in the alpha helix structure. Another secondary structure is beta sheets. These structure formed due to hydrogen bond formation in between the amino acids of the adjacent strand of a long polypeptide chain. The beta sheet can be of two types, parallel and antiparallel, with the different values of torsion angle between both these structure. The antiparallel structure of beta sheet is much more stable, as we can see from the diagram that the hydrogen bond in case of antiparallel is collinear, while it is not in case of parallel one. So these are physically much more stable as compared to that of parallel structure. Another secondary structure is beta turn, that is present mainly at band of protein and are rich in glycine and proline, as these two amino acids makes easier to bend for a polypeptide due to their special R group. Though these amino acids are usually not found within the structure of beta sheets and alpha helix, as their R group can destabilize the structure. In case of proline, there is a rigid ring is present that hinders the degree of rotation of dihedral angle. Thus, it cannot acquire a stable secondary structure, while if a polypeptide chain is rich in glycine, then also because of higher degree of freedom of rotation around the angle, make it an unstable structure. So, a stable structure of protein depends upon the degree of rotation of dihedral bonds, composition of polypeptide chain, that is what type of amino acids are there in the structure of protein. Bulk and shape of R group is also one of the most important factor, and the amount of proline and glycine is also contributing in the structure, in the stabilization of the structure. Suppose now if you have a unknown protein, then how can you determine their structural feature, and in a way that in what kind of conformation it is packed? This can be resolved with the help of Ramachandran plot. Here is we have a Ramachandran plot in which x-axis shows the will represent the phi angle, while the y-axis will represent the psi angle, and both are started with minus 180 to plus 180 degree. And the main purpose of this plot is to locate the phi and psi angle within the quadrant, and from the figure we can see that all the alpha helix are lying in this quadrant. So we can say that the alpha helix the phi angle and the psi angle are lying in a positive range, as the here is the phi angle showing their scale, while the psi angle are also occurring at the positive range, within the positive range, while in case of beta sheets it is not the same. The psi angles are showing their positive values, while the phi angles are within the negative values. So this is all about the primary, secondary structure and the Ramachandran plot. Ramachandran plot is basically helping us to determine the structure, that is the secondary structure of any protein. So in the next videos we will discuss about the tertiary and quaternary structure of protein. If you like this video, please hit the like button and mention your comments in the comment box, and please share it. Thank you.